Good afternoon. The article I chose from the popular media is titled RNA Molecules in Maternal Blood May Predict Pregnancies at Risk for Preeclampsia. This article immediately captured my attention because I'm very interested in maternal health. The scientific article titled Discovery and Verification of Extracellular miRNA Biomarkers for Non-Invasive Prediction of Preeclampsia in Asymptomatic Women was published in the Cell Reports magazine on May 18, 2020, in correspondence with the University of California, San Diego. The goal of the study was to identify an accurate method for identifying preeclampsia in women prior to birth. Since there currently is no accurate method of detection, preeclampsia often results in serious complications for both mom and baby. The study focuses on the small RNA sequencing of maternal serum among women who would later develop preeclampsia to look for indicators. So what is preeclampsia? Preeclampsia is a complication of pregnancy. Symptoms of preeclampsia include hypertension, protein in the mom's urine, sudden weight gain or swelling, severe headaches, abdominal pain, and/or nausea. Typically, these symptoms would arise late in the pregnancy or around the time of birth. Preeclampsia can develop into eclampsia, which is a very serious condition. Women that experience seizures have progressed from preeclampsia to eclampsia. Preeclampsia may also lead to death. It is estimated that preeclampsia is the cause of 15% of preterm births and 14% of maternal deaths worldwide. The only way to stop preeclampsia is to deliver the baby. Although complications of preeclampsia often arise late in pregnancy, preeclampsia actually begins developing very early on in the pregnancy. Preeclampsia begins in the placenta. When the placenta develops, new blood vessels are developed to send blood to the placenta. When these new blood vessels do not develop or function efficiently or properly, women have been shown to experience preeclampsia. Additionally, the blood vessels may not function properly because of pre-existing conditions in the mother, such as diabetes or high blood pressure. The development of effective prevention and treatment strategies for preeclampsia is limited by the lack of accurate methods for identification of at-risk pregnancies. So what are microRNA?
Every cell in our bodies has all our DNA, but clearly, not all of our genes are needed to make each individual part of our body. How, then, do our cells prevent unnecessary genes from making unnecessary proteins? Let's find out by taking a closer look at microRNA, which is a key ingredient in switching genes on and off. Like DNA, microRNA is created in the nucleus. Each unique microRNA sequence is encoded for by DNA using the ATCG base pairs. These segments are transcribed into an RNA sequence by RNA polymerase, which matches each base pair with its RNA complement. Unlike regular genes, however, the sequences that code for microRNA are found in introns, or non-coding regions of RNA. This means that they aren't translated into proteins. Instead, the resulting RNA sequence, also known as primary microRNA, gets processed by the dicer enzyme into its final single-stranded form, which is a mere 22 nucleotides long. The mature microRNA is then bound to an Argonaute protein, which is the secret sauce of genetic regulation. Now, microRNA is ready to get to work. MicroRNA regulates gene expression by RNA interference, also known as gene silencing. This happens after the transcription of DNA into RNA. In essence, microRNA prevents messenger RNA from translating or coding into proteins. Each microRNA sequence has a complementary messenger RNA sequence in that they have complementary base pairs that match up almost perfectly. This is how a microRNA molecule is able to find its target. In order to turn off a gene, a microRNA molecule will bind to the messenger RNA sequence of the gene that needs to be turned off by matching complementary base pairs. This process occurs with the help of the Argonaute protein, which lines up the microRNA for interaction with its target. Together, the Argonaute protein, dicer, and microRNA form the RNA-induced silencing complex, which acts like a multi-tool of gene silencing. By binding onto the target messenger RNA sequence, microRNA acts as a roadblock to stop the ribosome from translating at the site of binding. The Argonaute protein attached to the microRNA also has the power to cut the messenger RNA molecule into two pieces at that same site, effectively destroying the gene. As this destruction is occurring, the poly-A tail of the messenger RNA degrades, dealing the final death blow to the messenger RNA strand. The central dogma of biology has now been cut short just in time to stop a rogue gene from producing more proteins than a body needs.
So that kind of shows you a visual of microRNA and how they work. But just to sum that up, microRNA are small non-coding RNAs. They are typically 18 to 28 nucleotides long, and the main role of microRNA is the post-transcriptional regulation of gene expression. They regulate their target messenger RNAs, which results in the regulation of a multitude of cellular processes such as proliferation, differentiation, and apoptosis. Small non-coding RNAs also act in gene silencing. So microRNAs and preeclampsia are related. A biomarker is a biological molecule found in bodily fluids such as blood or in tissues that is a sign of a normal or abnormal process or of a condition or disease. Biomarkers have the potential to allow us to predict, diagnose, and/or monitor disease. Since the expression levels of certain microRNA are altered in different diseases, microRNA can be used as biomarkers. MicroRNA are also good biomarkers because they can be easily accessed in bodily fluids and they are easy to work with. There is significant evidence that various different types of cancer have unique microRNAs. For example, in the case of prostate cancer, one study done in 2015 was able to identify 14 different types of microRNA that were only present in the serum of patients with low-grade and not high-grade prostate cancer. MicroRNA have also been studied as biomarkers in other cases such as lung cancer and Alzheimer's. Since it was discovered that microRNAs are stable in circulation and that the placenta is capable of releasing microRNAs to the circulation in exosomes, scientists are enthusiastic about using microRNAs as biomarkers for preeclampsia. So the study that was done by collecting maternal serum samples at two different sites. One site was with the University of California, San Diego, and the other site was with Sera Prognostics, a biotechnology company that's based out of Salt Lake City. 48 samples were collected at the UCSD site, and 164 samples were collected at the Sera Prognostics site. These samples were then split into two categories, a discovery set of samples and a verification set. The discovery set was comprised of 141 subjects, and you can see that 49 of these subjects were preeclampsia cases and 92 were controls. And then in the verification cohort, there were 71 subjects and 24 of them were preeclampsia cases and 47 were controls. Among each of the categories, the subjects had matching gestational ages and disease severity, and also in each category you can see that there were about 2 times the number of controls as preeclampsia cases. So to analyze the maternal serum, small RNA sequencing was performed on the extracellular RNAs, and this was done in an effort to discover and verify different microRNAs that were expressed in subjects who then later developed preeclampsia. Each of the different cohorts, discovery and verification, were analyzed by this small RNA sequencing. RNA sequencing generates microRNA sequencing libraries from a complete set of RNA, and this is done in an effort to better understand the role of non-coding RNA. To better understand the non-coding RNA, small RNA sequencing isolates and sequences small RNA species, and this allows scientists to understand how post-transcriptional regulation can contribute to the phenotype And it can also allow scientists to identify biomarkers and capture the complete range of microRNA species. So overall, the study found that 2 single microRNA biomarkers, univariate, and 29 2 microRNA bivariate biomarkers were measured in the serum of asymptomatic pregnant women between 17 and 28 weeks of pregnancy were able to predict the later onset of preeclampsia. So this shows that the bivariate biomarkers were significantly more likely to predict the later onset of preeclampsia. The majority of the verified biomarkers contained MIR155-5P, and this has been reported to mediate the preeclampsia-associated repression of endothelial nitric oxide synthase by tumor necrosis factor alpha. And the study also revealed that several verified microRNA biomarkers came from the placenta and were likely carried by placenta-specific extracellular vesicles. Because of this, the study then speculates that extracellular microRNA biomarkers for preeclampsia may be indicators of placental or maternal tissue stress and serve as signaling molecules between the placenta and maternal tissues or between maternal tissues. And then since biomarkers are in tissues, this would make sense. So lastly, 3 of the extracellular microRNA biomarkers that were identified in this study are stated to have been previously associated with hypertension, and you can see these in the chart on the slide. So HSA MIR26B 5P and HSA MIR7 5P were found to be more present in the plasma of non-pregnant patients with hypertension than in normotensive patients. And then the HSA MIR26B 5P appeared to be mostly derived from the liver and the placenta. And then the second one, HSA MIR7 5P, was derived from the brain. And then the third microRNA, HSA MIR181A 5P, was shown to decrease blood pressure in hypertensive mice, and it was shown that this was derived from the placenta. So it's important to note that 2 of these microRNA are derived from the placenta, which plays a direct role in preeclampsia. And so overall, the study was able to verify several microRNA as biomarkers for preeclampsia, and this provides hope that a non-invasive detection of preeclampsia is on the rise. And then this is my works cited.